The future of humanity began in a lab decades ago. It was in that lab where scientists first unlocked the secrets of DNA, giving us the ability to understand life at its core. And in return, this discovery sparked a revolution. One that would allow us to not only read life's code, but rewrite it. The birth of genetic engineering set the stage for a new era, filled with incredible possibilities and profound ethical dilemmas. But what is genetic engineering? Simply put, it's the process of altering the DNA of living organisms, whether plants, animals, or humans, to create specific traits or solve problems. Unlike traditional breeding, which relies on natural selection, genetic engineering allows scientists to directly manipulate the genetic material to achieve desired outcomes. DNA, as we know, is the blueprint of life. It carries the information that tells cells how to grow, develop, and function. For years, scientists didn't fully understand how this information worked. But in the 1950s, two scientists, James Watson and Francis Crick, made a discovery that would change everything. In 1953, they figured out the structure of DNA, and suddenly, everything about life began to make sense.
Well, Francis, do you think we were lucky to have solved it, or was it real brainwork?
Well, I guess we were certainly lucky, and of course you give the impression in your book, Jim, we didn't really do too much thinking, but we were lucky, I think, for two reasons. We were thinking about the problem at the right time, and then the two of us, by collaborating, when one of us got on the wrong track, the other one could get us out of it. When if I thought there were three chains at one stage, you were sure there were two. If you thought that the phosphates had to be in the middle, then I would be the devil's advocate and say.
Watson and Crick's discovery showed that DNA is shaped like a twisted ladder, with the rungs of the ladder made up of pairs of molecules. This structure explained how genetic information is passed from parents to children. But while this discovery helped us understand how life works, it also opened the door to the possibility of changing life itself. If we could understood DNA, would we also change it? And what if we did, what would that mean for the world? In the years after this breakthrough, scientists around the world raced to understand more about DNA. They began to ask questions about how genes, specific pieces of DNA, control traits like eye color, height, and even susceptibility to certain diseases. The idea of changing genes started to take shape, but it wasn't until the 1970s that genetic engineering really took off. In 1973, two American scientists, Herbert Boyer and Stanley Cohen, made a discovery that set the stage for genetic engineering. They figured out how to move genes from one organism to another. They took a gene from one species and inserted it into the DNA of another species. This was the first time anyone had successfully altered the genetic code of an organism. With this new technique, scientists could now create genetically modified organisms, or GMOs. This wasn't just science fiction anymore. The idea of changing the DNA of plants, animals, and even humans was now a real possibility. And it didn't take long for scientists to start using these new techniques to solve real-world problems. In 1978, the first genetically engineered drug was produced, human insulin. Before this, people with diabetes relied on insulin that was taken from cows or pigs. Surprisingly, it worked, but it was expensive and in limited supply. With the new genetic engineering techniques, scientists could now create insulin in bacteria, which could be grown in large quantities. This was a game changer, making insulin much more affordable for people with diabetes. But as scientists started to make these changes to living things, some people began to worry. If we can change the genes of plants and animals, what's to stop us from changing the genes of humans? What would happen if we started playing God and altering life in ways we didn't fully understand? These questions started to swirl around genetic engineering as the technology began to grow. In the 1980s, genetically modified crops started to appear in fields. The first GM crop, a tobacco plant, was created to resist a virus. Soon after, genetically modified crops were designed to resist insects and tolerate herbicides. This was seen as a way to make farming more efficient and help feed the growing world population. But not everyone was convinced. Some people worried that these GM crops would crossbreed with wild plants, creating new problems. Would these new plants harm the environment? Could they end up in the food supply without anyone knowing? The 1990s brought even more advances in genetic engineering. In 1996, the world's first cloned mammal, Dolly the sheep, was born. Dolly was cloned from an adult sheep's cell, making her an exact genetic copy of the original sheep. This was a major achievement in genetic engineering, but it also raised even bigger ethical questions. Could scientists really clone humans? Should they? And what would that mean for the future of life on Earth? These questions were not easy to answer. At the same time, scientists were using genetic engineering in medicine. In 1990, the first successful gene therapy treatment took place. A young girl with a genetic disorder called bubble boy disease was treated with a healthy gene that fixed her immune system. Gene therapy offered hope for curing diseases that were caused by defective genes. But even as scientists were solving some of the world's toughest problems, the debate over the ethics of genetic engineering was just beginning. By the late 1990s and early 2000s, the Human Genome Project was underway. This international effort was aimed at mapping all the genes in human DNA. It was a massive task that would help scientists understand how genes control everything from our health to our physical traits. But the more scientists learned about the human genome, the more questions they had. As the century turned, genetic engineering was no longer just about growing better crops or producing medicines. The ability to alter DNA could, in theory, be used to change human traits. Scientists were starting to talk about the possibility of designer babies with specific genetic traits. Could we use genetic engineering to make babies smarter, taller, or more athletic? Should we even try? The potential was enormous, but so were the risks. Genetic engineering was no longer a distant possibility. It was a real and powerful tool, and the questions about how it should be used were becoming more urgent. Could we use it responsibly, or would it lead to a future that no one could predict? As the 21st century unfolded, genetic engineering entered a new and exciting phase. The greatest breakthrough came in 2012 with the discovery of a tool called CRISPR. Unlike earlier methods, which were slow and complicated, CRISPR allowed scientists to edit DNA quickly and precisely. It was a technology so powerful that it promised to change everything we knew about genetics. But with its potential came a new wave of ethical questions. CRISPR, short for clustered regularly interspaced short palindromic repeats, was initially discovered in bacteria, where it served as a kind of immune system, protecting the bacteria from viruses. Jennifer Doudna and Emmanuel Charpentier were the scientists who figured out how to harness CRISPR's ability to cut and modify DNA. They realized that they could use CRISPR to edit genes in living organisms, making precise changes with a level of accuracy that was previously unimaginable. In 2012, they published their discovery, and the world of genetic engineering would never be the same. CRISPR was revolutionary because it was simple, fast, and cheap. Scientists could now edit specific genes in a way that had never been possible before. This was a game changer for medicine. In theory, genetic diseases like sickle cell anemia or cystic fibrosis could be cured by editing the genes responsible for these conditions. And the possibilities didn't stop there. CRISPR also promised to help grow crops that were more resistant to disease or to create animals with desired traits, things that seemed like science fiction just a few years ago. But CRISPR wasn't just about fixing genetic diseases or improving crops. It raised a new and much more controversial question. Should we use CRISPR to change the human genome? This is where the ethical debate really took off. If we can fix genetic disorders, can we also enhance human traits? Could we make babies with better intelligence, improved health, or even physical traits like greater strength or beauty? If so, who gets to decide which traits are better, and who would have access to this technology? The first big ethical test of CRISPR came in 2018, when a Chinese scientist named He Jiankui announced that he had used CRISPR to edit the embryos of twin girls. His goal was to make them resistant to HIV, a disease that could have infected them through their father. The announcement shocked the world. Many scientists and ethicists condemned his actions, arguing that editing the human germline, genes that are passed down to future generations, was a step too far. What if the changes had unintended consequences that could harm the girls or future generations? He Jiankui's experiment caused an uproar. Many scientists argued that it was irresponsible to edit the human germline without fully understanding the risks. He Jiankui was eventually sentenced to prison for his actions, but his work sparked a global debate. While human gene editing remains a highly controversial subject, CRISPR has already proven itself in other areas. In agriculture, CRISPR has been used to create crops that are more resistant to diseases and pests, offering the potential to address some of the world's food security issues. It's also being used to produce better quality meat and livestock, which could lead to more sustainable farming practices. But even in these fields, the technology hasn't come without its critics. Some worry that genetically modified crops will disrupt ecosystems or create new environmental problems that we can't predict. In medicine, CRISPR has shown tremendous promise. Scientists are using it to treat genetic diseases like sickle cell anemia, a condition where a person's red blood cells are shaped abnormally, causing pain and other serious health issues. Clinical trials are now underway to test the effectiveness of CRISPR in treating such diseases by editing the patient's own genes. Early results have been promising, with some patients showing signs of improvement. But even with these successes, there are concerns about the long-term effects of gene editing. Could there be side effects we don't yet know about? And what happens when you make permanent changes to a person's DNA? Beyond medicine and agriculture, CRISPR has even been used in other fields, such as creating gene drives to control disease-carrying insects like mosquitoes. A gene drive is a system that spreads a specific genetic trait throughout a population at a much faster rate than traditional inheritance. This could help control the spread of diseases like malaria by altering mosquito populations to prevent them from transmitting the disease. While this technology could save millions of lives, it also raises concerns about unintended consequences in the environment. As CRISPR technology continues to advance, the ethical questions surrounding it grow more complex. Could we one day use CRISPR to enhance human intelligence, strength, or even physical appearance? If we have the ability to improve human traits, should we? And who decides what traits are improvements? These are questions that have no easy answers, and the debate is far from over. If you enjoyed watching this video, please leave a like, share, and comment your thoughts on this. Thank you for watching, and we'll see you next time.